Good afternoon, everyone. Two men, I guess, does not a panel make, so it is a dialogue. I want to put everything in a context for a while. I've been in the CMO spot. I've been throughout industry, both on the provider side, the taker side, and the giver side. But the CMO, and I'll include the clinical development team, is at a flashpoint in the clinical development program between patient resources, safety, and corporate goals. Everybody's been smacked in the face in this industry with do it cheaper, do it faster, and do it with less resources, which is an antithesis to quality, oftentimes, probably almost all the time. So one of the things that Jim and I are going to concentrate on is one part of that development program, and it's a critical part, and that is how you set up your program for the drug or product you're developing, and particularly how you set up the clinical trials within that program from a biostatistical standpoint. Now, obviously, we won't be able to give answers to exact questions because every program is different. Every program has its nuances, but we'll have you think about a process or a method to approach the problem. Before I start into that, though, I'd like to put up the next slide. Am I doing that? Okay. And some recent interesting findings in the literature about the p-value. Alive, dead, dying, misused, or all of the above. So I found a few quotes which are interesting, frightening sometimes. Let's look at the upper. Well, with p-value of 0.5 as a magic cutoff point, you are very likely to make a fool of yourself about 30% of the time. And if you're underpowered, you will be wrong most of the time. This was just published in the 2014 Royal Society of Medicine Open Journal by a guy named Dave Calhoun, who's thought about this for a while. PLOS Medicine. Most published research findings are false. A 1990 to 2015 15-year survey, 2 million abstracts and publications were looked at, 4.5 million p-values were reported. Almost all of them were significant. There should be a surprise right there. That's probably not probable. And only a small fraction of these included confidence intervals, Bayes factors, effect sizes, or other uncertainty metrics. The p-value. This is a great paper by Steve Goodman that appears in Seminars in Hematology in 2008. The dirty dozen misunderstandings about the p-value. Most ubiquitous, most misunderstood, most misinterpreted, and occasionally miscalculated index in all of biomedical research. Let's go to the Sunday Telegraph, a more lay approach to it. 70 years ago, Fisher gave us a mathematical machine to turn baloney into breakthroughs, flukes into funding. In its 177-year history, the American Statistical Association has never done anything like this, but recently they issued a warning about a procedure rather than a policy issue, and they said this was something about p-value misuse, and the statement aimed to halt missteps in the quest for certainty. And the last quote, which I think hits everyone right where it hurts in the wallet, is from 2015 Forbes. The single biggest determinants of a pharmaceutical return on investment are late-stage trial failures. For novel agents, this can rise to about 75%. We're all in the novel agent area, I would believe. The major reason, a fundamental misunderstanding of the humble p-value. So that's kind of a setup for Jim to go into the ins and outs of the p-value. Is it dead? Is it dying? Is it just misunderstood? But I think
p-values are not invalid. As the slide said, it's about misuse but more often misunderstanding. And so let's consider the key question for a trial. The more I do clinical trial consulting, the more I realize that p less than 0.05 is probably not the key question for the research. More important question is often, what's the probability of success for the drug? And usually the probability of success can be defined in some quantitative terms. And so we're going to look at that, at how to address the problem from both viewpoints with an example.
And before we get into that example, I want to look at a few things that can affect the probability of success. Recall, we're just looking at study design to biostatistical aspects of this, but you have to think about your drug's effect size, its true effect size. I've said many times that anyone could have developed GCSF, a cow could have developed it, because it was such a beautiful drug. Its physiologic action was its major side effect. You gave too much, you got too many neutrophils. It was a beautiful thing. We're not all that lucky, as we know that. We're often in the gray zone when we develop drugs. The power of this study. Do you have informative priors, or are you just getting into it and you don't have that blueprint? The measurement quality. Can you measure what you think you're measuring with any precision? A randomization procedure. The size of the clinical trial. Underpowered clinical trials are a major problem in the industry. The development program design. How does the clinical trial fit into the all-over development program, and are you asking the right questions? Study conduct. Do you, after the trial has been started, issue waivers? Do you allow for protocol deviations? Did you take the results from the prior trial and look at the sample size and the patient population and design the next trial exactly the same way? Or did you change a few things because you learned it? Well, you're changing your statistical assumptions along the way as well. Then there's the issue of bias. There's bias you can't control and there's bias you can control. For example, bias you may be able to control is if you go to a site and they have 3 clinical trials ongoing, they say, you've got some competition, your eligibility criteria are just about the same. Do they have a procedure for taking the patients, it was mentioned earlier in one of the speakers, and saying, okay, first patient in the door goes into trial A, second patient trial B, third patient trial C, fourth patient trial A, and do that? Or do they say, this one looks a little sicker, I think we're going to put him in the trial that I think they can tolerate? They've already introduced bias that you may not even know about unless you ask that at the time you're setting up your clinical trial. So all of these things influence your probability of success, and this is probably not a bad tick list to go through in your head when you're setting up your clinical development program or you're setting up an individual clinical trial. Say, am I asking the right questions about all the things that can influence my probability of success? How am I going to separate the wheat from the chaff and use my resources and dollars and spend them in the right direction?
And since statistics plays a large part in what we do in clinical trials, the earlier you start that dialogue, even before the IND, so that you can develop the IND, because the IND contains an outline of your development plan, the better off you'll be. And so let's carry through the example to make the points about the distinction between the p-value approach and probability of success. We start with a simple 2-arm trial. A control group response rate we know is about 40%. And we know that there's a competitor out there that has a drug that provides a 10% increment at least. And so we, in order to have marketing success, need to have at least a 15% increment over the control group to be successful. And so our criteria for success is the response rate for the test drug has to be at least 15%. And so if we power the trial in the usual way with 80%, and at the proof of concept stage, if we consider a one-sided alpha level of 0.05 and 80% power, we find that if the true underlying difference is 15%, we need 134 per arm. And that's just for proof of concept. So we realized, well, that's too large for proof of concept, and so let's think of some alternative approach. Well, we could be optimistic about our drug, and let's plan the trial for 20%. And that gets us down to 75 per arm for 80% power to yield a p less than 0.05. And then we also consider that that's for a true underlying difference of 20. And now we realize that 75 per arm has a minimum observed difference that'll be significant, and that's 14. So there's enough precision, in quotes, to see or observe a difference of 15. That means if we get 15, it's going to be significant. If we don't get 15, it's probably not significant.
So that's one of the things, Jim, is we don't know what the true underlying difference is. And how does the statistician figure that into a clinical trial design? I mean, nobody knows that, really, because
Right.
That's taking everybody in the world with that particular indication and treating them. So how do you do that?
It's about comparing the probability that you'll get the successful outcome in the trial, in this case 0.05, for various true values. We picked 20. If we wanted to be more rigorous, we would have picked 15, but we need more money for that. But we do know that if we get an observed value of 15, then that would be significant. So that's another way to look at the design. So the second approach is a decision analysis approach, and this involves the criteria for success of the drug. We just simply compute the probability of success, and that is the probability that the true difference exceeds the target. And in order to make a go decision from the trial, we're going to say we need at least 70% probability of success. But for a no-go, if the probability of success is less than 50%, we're going to abandon the program. But there's a gray zone in there between 50 and 70 where we're going to need more information about the drug. And in that case, we might look at secondary endpoints, do another trial, or do a sample size re-estimation to enlarge the trial. And we can incorporate prior information to the calculation of probability of success via Bayesian approach if we have some good prior information that we want to leverage. We could also consider an adaptive approach. And so now let's look at the performance characteristics of these two approaches for a spectrum of true underlying differences ranging from 10 to 40. And focus on the 20% line. We know we have 80% power in this trial to yield a p less than 0.05. And if there's a larger true difference, we have high power. If there's a smaller true difference, lower power. But when we consider the probability of success of success of the drug in the program without any prior information on the 20% line, we realize we only have 52% chance of yielding a go decision. But if we leverage prior information, that's equivalent to 75 per group. We can improve that only by 10%. So with a drug with a true underlying difference of 20%, we're only a little over 50% sure that we're going to be able to demonstrate success for the drug, but we're highly sure that we're going to get a p less than 0.05. So p less than 0.05 only means that the drug effect is bigger than 0. But that doesn't inform on the probability of success of the drug, which is an important factor in whether you move that drug forward or not.
So you'll still incorporate the p-value, but you will put something on it to put a little bit more truth behind it in terms of real clinical data.
For go, no-go decision making, yeah. So you could back off from the 70%. You know, that's an arbitrary number. It's the number that the company has to be comfortable with in moving forward after the trial.
Still, once you let this horse out of the barn, so to speak, you've committed dollars and resources, and you might not find this out until the end of the trial. So one of the things I think we can consider is the use of an interim analysis in that type of a design, and I'd like to go over that just a little bit here. In my view, and Jim and I have both dealt with the same clients and helped the same companies in this area, interim analyses can be helpful if you have a larger than affordable N. If the proper design of your trial calls for 150 patients, is there a way to get your cake and eat it too? And one of the things that an interim analysis or interim analyses can do is it's potentially good for news flow, because your CEO, your board of directors is down on you for saying, so I'm going to start the trial and I'm only going to get information in 2 years? Well, we can't do that. We can't commit that number of funds. We have to have some information as we go along other than we've completed enrollment. So it's potentially good for news flow if you come up with an interim analysis result that says we're going to continue the trial as is. It can also help allocate budget spend. If you go into a clinical trial and you say after the first X number of patients, we're going to take a good hard look at the data in a robust statistical fashion and make the decision based on the money we have or the money we can raise whether or not we should go forward, or whether we're going to say, no, this is too risky, we're going to allocate our resources somewhere else. Increasingly, I have seen that IRBs, ethics committees, PIs, particularly at academic institutions, are pushing back and saying, even in phase 1, 1B trials, where's your statistical analysis plan? You're doing an expansion phase for 20 people, but you have no method of saying how you're going to analyze that data. We are reluctant to commit ourselves and the patient resources to that, so we kind of have to put them in. So I think as Jim and I talked about, there are several different ways to skin the cat here, but we'll talk about one, which is called the beta spend function. And I think you can see that you can have your cake and eat it too, but it comes with a little bit of a cost.
And that involves putting interim analysis for futility in. You don't take any alpha penalty when you look at the data at interim analyses for futility. So if we start with the example trial at 75 per arm, that had 80% power for that 20% difference. If we put in one interim analysis for futility, we reduce the power by a couple of percent because we're introducing another chance to stop for futility. And then if we introduce a second interim, it goes down a little bit further, a third a little bit further. And that's for a conservative beta spending, type 2 error, false negative spending function. And again, there's no alpha adjustment when you do only futility stopping.
So I think you can see from the to and fro between Jim and I, for those of you who speak stat speak, you can probably understand this. For those of you who don't speak stat speak, which I would guess is you're either a liar or you say you can't, because I can't. I've tried, rejecting the null hypothesis. Reminds me of a board question I had when I took my internal medicine boards, and I pitied the people who had English as a second language because the question read something like, which of the following is not true for pseudo-hypoparathyroidism? I just skipped that question and went on to the next one because it was just too long to think about that. It twisted my brain. So
it was A and C, wasn't
it? A and C, yeah. I'm sure it was. So when you involve a biostatistician, I think Jim has alluded to this earlier, as soon as you can get your hands on your drug program and you have some money and you want to go forward at the pre-IND stage, because you got to start thinking about all these things that determine your probability of success. You have to learn the language about your product, but you have to learn about the statistics of your product and your development plan. So I think that, at least in my experience, having frequent talks with biostatisticians about clinical trials has been exquisitely helpful in understanding what you're getting into and then being able to translate to the people who need to know, like your CEOs and the people who are paying your paycheck, what that means to them. You can't be certain, there's the seductive certainty of significance, but at least going into the program with your eyes open, what kind of risk you're getting into. So I think we'll stop there and take any questions, because then that involves the next part, which is a whole different form of bias, interpreting and reporting your results. Catherine.
So I had a question. Back in the old days when I was at AZ, we had a plan to do randomized phase 2 trials, and instead of using a p-value of 0.05, we used a p-value of 0.2. And the idea was that we knew it was quote, underpowered, but at least it would give us an idea of how to estimate how likely we were to be right or wrong about the hypothesis, and that would inform decision-making going into phase 3 as sort of a compromise position. What do you think about that?
Yeah, and that's because you were in the hypothesis testing power calculation framework, which is p.2 instead of p.5. So you could have used the calculation of probability of success and relaxed that probability that you need.
And generating informative priors.
So, hi, I'm Shamim Ruff from Sarepta Therapeutics. So you mentioned futility analyses, and I think that's fine. You want to check that this study is not going to be futile, and you want to save money if it's not going to be positive. But can you talk a little bit about early designing studies for potentially early efficacy reads? And the reason I ask this is certainly for in the DMD space, we're looking at a study that's going to be 2 years in duration. And a lot of the patients and patient groups are reluctant to go on a placebo-controlled study that is 2 years long. So they genuinely believe in the drugs or the platform drugs that we're working on, and we have to demonstrate that we have a good interim analysis. Obviously, it will be under an IDMC, et cetera, but that's not going to cost too much in terms of alpha, but still be realistic in terms of seeing efficacy if it actually exists after a year. Could you talk a little bit about that?
Yeah, I mean, there are methods in place to accommodate interim analyses and spend very little alpha so you don't impact the sample size, but if results are robust, you end up getting to stop early anyway. And they can be calculated and applied for each situation, depending on what your logistical constraints are. Bayesian approaches are acceptable to regulatory agencies with the same kinds of false positive rates that need to be investigated, often via simulation instead of closed-form calculations. But all of that can be done. And optimal methods for each situation can be found.
And then I think the other important aspect of taking these into account for a clinical trial group is interpreting that in by itself in a vacuum is not a good idea. If you've read all these statistical literature things that I have read, just taking that sometimes does you a disservice. You have to look at it in toto. So take the clinical data, take some of the things you really can't measure or aren't measuring at the time, as well as the safety risk profile, put those in together. And you can put language into a protocol which says, we'll do a futility analysis, but that doesn't mean we're definitely going to stop. We're going to look at that and consider it in light of other clinical information, safety information, then make the decision whether or not to go on. And that's usually acceptable to folks who are reviewing your protocols.
And as you probably know, in the context of a pivotal trial, blinding, firewalls, and those sorts of issues need to be addressed.
Hi, thanks. So the good news in this crowd, I think, is our statistician colleagues like you, Jim, and John, and maybe some others would agree that the medical folks never back into sample sizes, right, based on how much money we have or how many people we think are reasonable to study, right? But I really appreciated your comment on a slide about the p-value is not the be-all and end-all, right? And what you're really looking for is evidence of probability of success to move forward. So I'll just give you an example. So I have a friend who's developing a drug in pancreatic cancer in phase 2. Now the design of the study for phase 2 and phase 3 is going to be the exact same design, right? Same endpoint. We're looking at my friend is looking at overall survival. And we powered the study to look for a 50% improvement in overall survival, right? Which is very ambitious in this disease, but that's around the size that most people are running phase 2 trials. It could well be that we won't get a p-value. We might see a 30 or 40% improvement, which is very robust. Now, if that happens, what's going to happen, right, is externally, our study failed because we didn't hit a p-value. And that's just, I mean, that's the way it is, right? So I don't know if you have any suggestions or thoughts on how to communicate, because I think your message here is absolutely right. But I think we collectively we could do a better job in trying to communicate what these results could mean.
Yeah, and so at the design stage, you could say if the true effect is 20% or 30%, we're going to come out of the trial with, I don't know, 80 or 90% probability that the drug effect exceeds our minimum clinically important difference. You can design your trial for that criteria rather than for a p-value criteria up front. Because it's phase 2, it's not for a regulatory filing. But if it's the same design, the same endpoint, the other thing you could consider is using that as a informative prior on the phase 3, things like that. But the answer is to design your trial with the criteria that you're going to need to make that statement to the external world when the trial is over. Design your trial for that statement rather than a p-value. In big pharma, I know of a couple of examples that are moving to that for go, no-go decisions along the way.
Looks like there are no other questions. We've got 3 zeros over here. I don't know if that's our rating from the judges, but we'll stop. Yeah, the red light's not on, right. Is that the median or the mean rating that we got? Thanks, everybody.